Hello, my name is Jim Blando, and I'm an associate professor at Old Dominion University located in Norfolk, Virginia. Today, I'm going to discuss an overview of the exposure assessment process, and in this section in particular, I'm going to focus on reconstruction of exposure. How we utilize computer models to help us reconstruct exposures, oftentimes to support retrospective epidemiologic investigations. Reconstruction of exposure is essentially where we utilize models to estimate what exposures may have been in the past. This can be very valuable to support retrospective epidemiologic studies, but this has also been utilized, for example, in legal proceedings in court cases, where someone may be estimating what the exposures may have been and what the attributable risk might have been for a given product or occupational environment, which is utilized in legal proceedings in terms of assessing responsibility between a plaintiff and a defendant. So reconstruction of exposure is often utilized for various purposes. So what is reconstruction of exposure? Reconstruction of exposure is essentially where I'm trying to estimate what exposures may have been in the past. It's very similar to the EPA approach of scenario evaluation, but in this case, it's retrospective. We're trying to assess exposures that may have occurred in the past. This relies both on modeling and also any pre-existing exposure data that may exist. This example here, this is a very interesting report cited on this slide. This is an example where in the Toms River Pediatric Cancer Study, the Agency for Toxic Substances and Disease Registry essentially retrospectively reconstructed the water distribution system in Toms River, New Jersey, and was able to utilize modeling to estimate for individuals living in this area what their exposures were through time to various contaminated water sources. And that's what this report was able to do, was able to ascribe past exposures to different water sources as the community and the water distribution system developed over a multi-decade period of time. And this was very valuable in terms of the epidemiologists being able to reconstruct the study to estimate what the risks may have been to people living in this community, which was a community that had elevated childhood cancer rates. And that was the purpose of the epidemiologic study. And this is how past exposure modeling or reconstruction of exposure contributed to that effort in that health study. Here's an example. I know this slide is very busy. The purpose of me putting these charts and tables up here is not really for you to gain anything out of it other than just make the point that, in fact, this is an example in the Toms River retrospective childhood cancer case control study where we were able to utilize that water modeling to help us assess what the risks were or the probabilities of children with cancer from having certain drinking water sources from certain contaminated sources in the community. So allowed us to model and estimate what exposures may have been and ascribe the probability or risk of a cancer case from having a given water source as their drinking water source. And it produced a lot of very valuable data, which was very helpful with that study. But this is where that retrospective methodology was utilized in an example of that. In addition to utilizing computer models, sometimes we can utilize biomarkers as well, especially if the biomarker is persistent in the human body. Depending on when you've got involved in this field, some of you may remember this as a field oftentimes referred to as molecular epidemiology. Today, a lot of folks tend to refer to this as omics, which is essentially utilizing biologic measures to assess exposures. Can be very, very helpful, has improved over the years and recent decades because of the expansion and increase in biotechnology and molecular biology. It's really, quite frankly, amazing to see what we're able to do these days with the biotechnology that's available to us in terms of measuring biomarkers in humans. Of course, because we are measuring these in humans, it can be very heavily impacted by host factors. Here's the US FDA definition of a biomarker, a little bit more broadly defined. In the old days, we oftentimes referred to as biomarkers as a blood or urine measurement, some type of body fluid measuring a chemical in the blood or urine as a way to assess what somebody's exposure may have been. Nowadays, because of these enhancements in biotechnology, we can look at much more sophisticated technologies, molecular techniques for us to measure many different things in the body, in body fluids. And that has really enhanced our ability to utilize biomarkers, especially biomarkers that persist in the human body, because that allows us to estimate what exposures may have been in the past. For human studies related to environmental exposure, biomarkers are often measurements of chemicals or metabolites in the blood or urine, as I've already mentioned. Sometimes in the past, people might use things like hair or fingernails because this might be a marker of a longer-term exposure or an exposure that happened far off in the past. This is oftentimes referred to as markers of exposure. Recent research has started to focus on biologic markers of effect. where researchers can look at things like your body's response to exposure and utilize that as a measure of exposure and to assess exposure. For example, if I see changes in protein expression, changes in RNA transcription, changes in microRNA expression, changes in gene expression, that could be utilized as a marker of effect of exposure, which also can help us assess what the potential risk is for a given exposure and help us assess what the exposures may have been. For example, we currently have a study going on where we're looking at microRNA expression in firefighters after they've been exposed to toxic combustion byproducts. So this is something that's developing now and likely will continue to become more and more useful in the future. Biomarkers can be used to reconstruct or estimate exposures. Again, one of the really classic examples would be blood lead. Very simple, inexpensive blood lead test. That may tell me what somebody's exposure may have been in the last 30 days. If I needed to go back and look a little bit further and say, did this person have significant lead exposure in the last 90 days, I might use something like the zinc protoporphyrin test, which can help tell me something about a little bit longer term exposure. That's kind of a classic example where I might be able to use a biomarker to help me reconstruct exposures. Of course, one of the problems with biomarkers has always been sometimes they're not very practical. It can be difficult to get people to agree to biologic measurements. Most people don't like getting stuck with needles. So, for example, if you're trying to draw blood from people, most people are not excited to volunteer for that. So it can be difficult to get those samples. May be difficult to reach a specific people that you're interested in assessing exposure on. And of course, one of the really important that sometimes is forgotten, quite frankly, toxicologic parameters is we have to consider the toxicokinetics, the pharmacokinetics of whatever chemical we're looking at. It's important to keep in mind that if a chemical has a short half-life in the body, a biomarker may not detect that in the biologic sample. And if it can't detect it, it might not indicate that there was an exposure present. So for example, if I think about inorganic arsenic, it has a very short half-life in the human body in blood, right? So if the half-life is, say, roughly 4 hours in blood, but somebody's exposure was several days ago, I could take a blood sample, and that blood sample will not tell me whether there was exposure to inorganic arsenic or not, because it's already been eliminated from the body by the time I collected my biologic sample. So that is one of the limitations we have to be mindful of when we're utilizing biomarkers. We have to consider the toxicokinetics of the individual chemicals in the body. Otherwise, we may get erroneous results when we utilize biomarker sampling. Getting back to that classic example with lead. I can't tell you how many times I've had obstetricians ask about, I have a patient who may have been exposed to lead-based paint from a home they lived in as a child, but now 20 years later, I ran a blood lead test. Is that a good marker if she had exposure in the past to lead? The answer would be no, because it's so long ago that the pre-existing lead in the woman's body would likely be stored in bone and would not exist in her blood at that time many years later, right? So again, the kinetics can have a very important impact on whether biomarkers are useful or not. This is just an example of a lead acid battery manufacturing facility. We had a lot of folks who had very elevated blood leads. This is just an example, again, where we can utilize biomarkers to tell us something about what exposures people may have. As you may know, if you're familiar with the lead in manufacturing standard at OSHA, we can actually medically remove people from the workplace if their blood lead gets above a certain value. But I can also go back and utilize these blood lead measurements to go back and estimate what the airborne exposures may have been and whether there's a violation of the permissible exposure limit as well. So to summarize, exposure reconstruction is often used when we have a need for retrospective assessment, like a retrospective epidemiologic study or a legal court case where a claimant is claiming that exposures they had in the past made them sick today. It heavily utilizes computer modeling, but may also utilize pre-existing sampling data. But biomarkers can also be utilized to estimate past exposures, as we've discussed. And because the technology has improved so much, we can also not only measure markers of exposure, but we can also measure markers of effect.